Okay, perfect. So thank you so much for that introduction. And I'm really happy to be here. And yes, I'm going to talk to you a little bit today about gut syndrome in horses. This is a topic that hasn't really garnered a lot of attention really over the last few years. Certainly not in horses, but in other species, particularly humans, we start to see a relationship between the level of activity and maybe more specifically the level of stress that the athletes are under. This can actually contribute to a very low grade inflammatory situation, which can manifest in a number of different ways, right? We see leaky gut syndrome in humans being associated with things like problems with muscle metabolism, chronic inflammatory diseases, for example, like arthritis, as well as many allergies have also been linked to leaky gut syndrome in humans. Now, we don't have a lot of data on leaky gut syndrome in horses, but we are, we're certainly starting to see that there's a relationship between what we see in human athletes and what we maybe are starting to see in equine athletes. So what I'm going to talk to you a little bit about today is just give you a little bit of background on what leaky gut syndrome actually is. And then near the end of the talk, we'll talk a little bit about some research that has been done in my research laboratory in Canada to start to maybe get a little bit more information about leaky gut syndrome in horses and hopefully start a broader discussion about how we can perhaps help some of these equine athletes by reducing the impact of leaky gut syndrome in horses. So we start at the beginning, right? The digestive system, certainly the people on this call and many people in the broader context of equine nutrition, we tend to sort of think a lot about the digestive system, and we think about it from simply the perspective that this is how the horse gets nutrients. But of course, there's a number of other roles that the digestive system plays. Absorption of nutrients is, of course, one of them, but the gut also has a really important job in terms of separating out the beneficial impact or the beneficial components of the diet from the non-beneficial components. So there is a sequestering of non-beneficial, non-digestible, perhaps toxic components of the feed, and that those components remain in the GI tract and ultimately come out the other end. So a really critical role of the GI tract is to maintain a selective separation between the feed that the horse consumes and the part of that feed that the horse wants to absorb. So this idea of egestion or getting rid of waste and toxins is a really important part of the GI tract. And for the GI tract to be able to perform that role, it has to have an intact, selectively permeable membrane. So that membrane is really, when we start talking about leaky gut syndrome, it's that membrane that starts to some extent break down. And so when that happens, some of these potentially toxic compounds can leak out of the gut and ultimately move into the blood. So egestion of waste and toxins is really important. The gut also plays a pretty critical role in hormonal homeostasis. So this is really the hormonal regulation of things like appetite. So for instance, the seeking behavior where the horse is actually looking for food, as well as satiety. So this is the behavior of the horse to be able to regulate his own feed intake. So we think about a horse, for example, who is sedentary. Those types of horses typically are eating about 2% of their body weight per day. But it is partly due to hormonal homeostasis in these animals that allow them to regulate what's actually going to be taken in. And part of that also, of course, is regulating metabolism. So we these horses are essentially listening to cues from the GI tract to tell them when they should eat, when they should stop eating, and then ultimately what they're going to do with the feed once it's absorbed. Another really important role of the digestive system is as a role in host defense. So we know that there is a vast amount of immune function that goes on in the GI tract. And a lot of that is also integrated in that semi-permeable gastrointestinal barrier. Now, one of the things that comes up more and more the older I get is this idea of the microbiome. So the microbiome is this very rich, diverse population of compounds in the GI tract of the horse, and these compounds that are in the gut, not only are they helping in terms of digesting or breaking down foodstuffs that the horse would not otherwise be able to digest, but there is a very complex relationship between the microbiome of the horse and the behavior of the horse. So we have a lot of very interesting relationships there. I'm going to talk more about the microbiome once we get into the next few slides. All right, so we'll start off a little bit talking about how some of these nutrients are absorbed. And this is really important. I'm not going to get into a huge amount of detail, but I want to point out a few things that are quite key to the discussion that we're having today. So you notice on the sort of cartoon picture there on the left that we have the lining of the GI tract that has multiple folds. And the purpose of these multiple folds is so that the horse can maximize surface area for absorption of nutrients. And if you look over on the right, there we have a picture of a single villi. And you can see within that single villi, there is a rich network of capillaries. So this is essentially how the nutrients that are absorbed from the gut move almost directly into the bloodstream. So if we're talking about beneficial nutrients, then this is obviously a very good thing. It's a very useful strategy to be able to get immediate benefit from nutrients that are being absorbed from the GI tract. The problem is when we have a little bit of a breakdown of barrier function, then this rich network of capillaries allows ready access of toxins directly to the systemic circulation. And this is where we potentially could have a problem. So if we take a little closer look in terms of the intestinal barrier, what this picture here is showing are the individual cells of the gastrointestinal lining. So these cells are called enterocytes, and these are essentially the cells that are regulating what gets absorbed to what extent. So if you look to the far left, what you're seeing there is that there are some large peptides and some proteins that can actually be taken up directly through active transport or passive transport directly through the cell. So this is through endocytosis as well as transcytosis. And many of the larger compounds that are going to be absorbed from the GI tract are absorbed through the enterocytes in this manner. Now, if you move a little bit to your right, you can see that there's a sort of a narrow blue line there, and that narrow blue line is called the pore pathway. So this is a pathway where small compounds, so things like water, for example, can get absorbed through the pore pathway, as well as many electrolytes get absorbed this way, and some small solutes. So you can see there's a sort of a little blue squiggle there at the top, and this little blue squiggle is very much like a gatekeeper. So these are called tight junctions, and these tight junctions are made up of a number of different proteins, and these proteins can change their shape a little bit to make the pore either a little bit larger or a little bit smaller. So these gatekeepers are highly regulated, and in a healthy GI tract, that is the major mechanism whereby small compounds can get from the lumen of the GI tract into the blood. Now, if we move now to your far right, and you can see there's a much thicker line here, and this is the line that we call the leak pathway. So this leak pathway is also regulated by tight junctions. But the problem becomes when these tight junctions, when their shape changes in a way that allows larger compounds to move through. Now, this is potentially, not always, but potentially a pathogenic pathway, because not only are we potentially moving in some large peptides, but you can also move through some bacterial products. So an example of a typical bacterial product that might get absorbed through this leak pathway is lipopolysaccharide. So lipopolysaccharide is found on the outside of bacterial cells, and in normal, healthy horses, we have a very low blood concentration of lipopolysaccharide, or we call it LPS. And there's good reason for that, because when you start to increase the level of LPS in blood, it triggers an inflammatory response. So that inflammatory response can be categorized by, for example, increases in compounds such as serum amyloid A, which is an acute phase protein that's made primarily by the liver. And we see when there's an increase in leakage of LPS from the lumen of the GI tract into the blood, then that is often associated with an increase in these acute phase proteins. And this is potentially not a good thing for the horse, because in low levels, it may not be a clinical type situation, but in larger quantities, then this can be really quite problematic for the horse. So the gut is the largest interface in the body that's in contact with the external environment. And so for that reason, it has a really key role in terms of distinguishing between the antigens that it should tolerate and the antigens that it should not tolerate. So this is part of the idea of host defense. And so again, when this system breaks down a little bit, then this can be potentially problematic for the horse. So I mentioned earlier about the role of the microbiome, and the microbiome is increasingly being understood as something that, well, obviously the horse cannot live without. This is a great example of a symbiotic relationship whereby the horse needs the microbes in order to digest food as well as other things, but is also, we also have an important role to play in terms of protecting the lining of the GI tract, and I'm going to talk about that in a second. So the microbiome has a key role in terms of development and function of the enteric nervous system. So what I mean by this is it's playing a key role in terms of gastrointestinal transit. So again, this is a key role of the microbiome. The microbiome is also producing a number of metabolites, and part of this metabolite story is the microbiome is directly providing nutrients for enterocytes. So nutrition for enterocytes, these compounds also may have some anti-inflammatory properties, and importantly, they fortify barrier function. So this, what these compounds are doing is they're enhancing epithelial cell function so that translocation of toxins through the leak pathway is reduced as much as possible. So the microbiome is also contributing to increased mucus production in the GI tract and has a key role in terms of regulating assembly of tight junction proteins. Now, this can go both ways, right? So one of the ways is that it can actually protect beneficial effects of tight junction proteins to make sure that sequestering of toxins remains within the lumen of the GI tract. But when you have situations of chronic stress, and of course, exercise is one of the key chronic stresses that our horses are being exposed to, one of the things that will happen is the microbiome will cease to metabolize a certain compound, and this compound is called phenylalanine. And this compound has been shown in humans to basically break down function of these tight junction proteins, which can increase leaky gut syndrome in humans. We don't have that evidence in horses, but it certainly seems a plausible explanation as to why exercise, and more specifically training, can have a negative impact on leaky gut syndrome. So what happens when we have this leaky gut? What are some of the implications of that? So we do get leakage of toxic molecules that can be too large to pass through the pore pathway, but they're getting through via this leak pathway. And when that happens, this can trigger a low grade, but certainly a persistent systemic inflammatory response. So some of the effects of this that we see in non-equine species are things like impaired muscle metabolism. And of course, if you're talking about athletes, this is going to have a negative impact on performance, as well as things like metabolic dysregulation, as well as allergies and some inflammatory disorders. So what makes a gut leaky? Well, I've mentioned this now a couple of times. Exercise is one potential contributor, as well as fitness. And these are actually contributing in separate ways, but they certainly seem to work together to potentially increase leaky gut syndrome in horses. And it's not just exercise stress, right? There are a number of other types of physiological stress, such as, for example, horses that are chronically, spending a lot of time in stalls, for example, that can really contribute to psychological stress. And diet, diet, of course, will impact leaky gut in both directions. Right? If you have a horse that is very stressed and maybe undergoing a lot of exercise stress, you can modify the negative impacts of that stress by modifying nutrition. And I'm going to give you an example of that near the end of our talk. So why does exercise contribute to leaky gut? Well, there are a lot of different theories as to why this happens, but a key part of that is that exercise results in diversion of blood away from the gastrointestinal tissues and ultimately to contracting skeletal muscle. So when you have that diversion of blood away from the GI tract, of course, what that also does is it diverts oxygen away. So when that's happening, you are potentially having a type of ischemic type situation that's going on with the GI tract. And that can have negative impacts in terms of metabolism of the enterocytes in the GI tract. Exercise also disrupts normal distribution of ions. Most of us are, or many of us are very familiar with the fact that ions play key roles in terms of the contraction relaxation cycle in muscle. So when you have these horses exercising at high intensity, that can have a major impact on ion distribution, both through the intracellular and extracellular fluid volume. And that can also have an impact on the GI tract. So we also know that exercise can contribute, because of this diversion of blood away from the GI tract, can contribute to swelling of intestinal cells. And that swelling can contribute to local inflammation, which again will further restrict blood flow to the enterocytes in the GI tract. So that's a potential contributor to that relationship that we see between exercise and development of leaky gut. Now, what's the impact of fitness? And this is a very new question, to the extent that, to my knowledge, no one has ever studied this in horses. But we do know that in humans and in some lab animals, we know that leaky gut syndrome in general and gastrointestinal disorders specifically are positively associated with increasing fitness. So we see this positive correlation between not just the duration and intensity of the exercise bout, but also the fitness of the individual. So the more fit the individual, the more prevalent these leaky gut clinical signs become. One of the pieces of evidence that we have for this is that GI disturbances are reported 3 times more frequently in elite athletes compared with amateur athletes. So this particular study was looking at, I mean, it's difficult to normalize an exercise bout between elite athletes and amateur athletes, but in as much as it's possible, a semi-normalized exercise bout was much more impactful in terms of gastrointestinal consequences in the elite athletes versus the amateurs. Now, the other really key piece here is that we know that there is a fairly profound impact of fitness on the gastrointestinal microbiome. And this goes back to the discussion I had a few minutes ago about ethanolamine, and that as a human gets more fit, we see an accumulation of this ethanolamine in the GI tract. And there are certain microbes that preferentially metabolize this compound. And unfortunately, these microbes tend to be the ones that are more pathogenic. So things like Clostridia, for example, E. coli, for example, these ones preferentially can metabolize ethanolamine, and that has the impact to destabilize tight junction proteins. So that's a potential mechanism whereby we see this relationship between increasing fitness and increasing incidence of leaky gut syndrome. So some of the effects of stress, so going kind of beyond just exercise stress and talking now more about stress in general. So we know that cortisol will increase during stress, and there is a clear relationship between the compromise of tight junction proteins between those intestinal cells being associated with increasing levels of cortisol. And so in animal studies, in rodents in particular, we see that there is an increase in gastrointestinal permeability in rodents that are experiencing chronic stress. And this was measured by increasing levels of cortisol. We also know that physiological stress will disturb the gastrointestinal microbiome. And I think we've made this point that there is that direct relationship between a healthy microbiome and a healthy tight junction complex. So when you have increasing amounts of physiological stress, then that will have the effect to increase gastrointestinal permeability. Now, I mentioned now a few times that we have almost no research here in horses. There was a study, this was actually a PhD thesis, can't remember, maybe it was an MSc thesis, but a thesis nonetheless out of Michigan State University in the US. And that study looked at the effects of things like phenylbutazone. So this is a very common anti-inflammatory drug that's used in horses. And that study clearly showed that administration of phenylbutazone increases gastrointestinal permeability in equids. So that is one piece of evidence that we do have in horses. So what do we know about the effects of diet? Well, in horses, we know very, very little. But we do have evidence in transported cattle that when there is certain supplements, now in this case, it was a molasses-based liquid supplement, was able to increase ruminal butyrate, and that decreased gastrointestinal hyperpermeability that was resulting from transport stress. So again, there's very likely a role here to play in terms of the microbiome. But that certainly gives some evidence for the fact that we can alter the negative effects of stress by altering the diet. So the next point there is, there was a study here in Holstein cows, and in those cows, they actually were able to increase gastrointestinal permeability by increasing heat stress in these horses. Now, there was an antioxidant supplement that these researchers were studying, and in this case, there was no effect. But we certainly have some pretty good evidence that heat stress is another way that animals will potentially develop this leaky gut syndrome. Another part of the diet, this is more from a negative perspective, is that we know that if we feed a high sugar diet to rodents, that will increase gastrointestinal permeability. Now, this was a really interesting study, because what these authors did is they treated some of these mice with dietary garlic, which was actually able to completely block the sugar-induced increase in hyperpermeability in these mice. So pretty cool. But that really makes the point that not only can we adversely affect the gastrointestinal barrier function with diet, but we can beneficially impact it also. There's also a kind of a really interesting question about what is the role to play of things like prebiotics and potentially probiotics or even postbiotics. If the gastrointestinal microbiome is playing such a key role in protecting barrier function, well, what happens if we can beneficially impact that microbiome through things like prebiotics, for example? Then I think that's a really interesting area of research that deserves some attention. So what do we have in horses? Well, not very much, I'm sorry to say. This is the thesis that I mentioned earlier. The author is Comper, and it was published in 2013. And what that study pretty clearly showed in horses is that gastrointestinal ulcers and phenylbutazone, both of them, will increase gastrointestinal permeability. There was a really nice review paper that came out a few years ago. This was published, I want to say it was out of North Carolina State. And this was a really great review in terms of the potential implications of leaky gut syndrome in horses, but it also made the point that there's really no horse research out there at that time. So there's not a lot else. So I guess a couple of years ago, I had a master's student in my lab who was really interested in leaky gut. So we ran a couple of studies. First of all, trying to develop a model for leaky gut in horses, and then seeing if we could modify the impact of that model by changing the diet. So the purpose of this study was really to create a model of equine leaky gut syndrome using a kind of a situation that was typical of a competition day type scenario. So essentially, we wanted to put a horse on a trailer, drive them around for a bit, and then once they got to their destination, then they exercised at moderate intensity for about 30 minutes. So the idea was to use a stressor that is typical of what a horse might encounter in normal everyday horse life. So essentially what we did is right before we put these horses on a trailer, we dosed them with a compound called iohexol. Now, iohexol is really interesting because it's actually a contrast agent that's used in pediatric medicine, and it's the molecular size of iohexol is just a little bit too big to fit through the pore pathway. So, but it's only a little bit too big. So therefore, if we compromise tight junction proteins a bit, then what we should see is we should see iohexol leaking out of the gut and moving into the blood where we can measure it. So we dosed all of our horses with iohexol, just an oral intubation of iohexol. And then we took a blood sample right before that. And then we put them on a trailer and drove them around for 60 minutes. And then we took another blood sample. And then the horse exercised at moderate intensity, just on the ground with lunging exercise. And then we took another blood sample immediately after exercise. And then we took more blood samples at 1 hour, 2 hour, 4 hours, and 8 hours post-exercise. Now, at the same time that we were taking these blood samples from these transport stressed and then exercise stressed horses, we also took blood samples from a sedentary control horse that was not exercised. So I'm going to show you some data now to kind of see what happened. So what you're looking at in this graph, this is blood levels or plasma levels of iohexol on the y-axis. And then along the x-axis, we've got the time points where we took blood samples. Now you can see between P1 and P2, so basically P1 is right before transport and P2 is right after. And the open circles are the horses that were transported, and the closed ones are the horses that were controls. And I'm sure most of you are saying, well, Wendy, they both went up. Well, yeah, they did. And the reason for that is because the horses we used for this study were, they were our research horses that normally live outside in a field. And we put them inside a stall for the purposes of this blood collection. So they were not happy about this. So I think this makes the point that even moving into a stall, if a horse is not used to that, can be a very stressful event. But look what happened at P3. So this is right after exercise. And you can see a big increase here in the uptake of iohexol, dare I say, the leakage of iohexol out of the GI tract and into the blood. So this makes a very clear statement about the effect of exercise on the permeability of the gut. And you can see then after 1 hour, 2 hours, and so on, we start to see that decline in iohexol over time. So some pretty interesting data, but they probably don't mean anything if we don't have evidence of a subsequent inflammatory response. And we do. So what you're looking at here is on the y-axis is a serum amyloid A. That's that acute phase protein I mentioned earlier. And then on the left, we have the control horses. So their serum amyloid A remained very, very low. Whereas the exercise stressed horses, now there was a wide variability here, but this is statistically different than the control horses. So this is evidence of an acute phase inflammatory response. And, you know, what caused that inflammatory response? Well, one of the questions is, did we have an increase in leakage of LPS from the GI tract into the blood? And at this point here, P5, we do have an increase in LPS in the exercise stressed horses compared with the control. Now, I do want to make the point that normal levels of LPS in blood is just under 2 micrograms per mil. So we're not really outside of the normal reference interval. So I don't think we should get overly excited about these data in terms of over-interpreting them. But I do think that it gives us some evidence that there is a relationship here between the stress and then also leakage of LPS from the GI tract. So then the idea was, well, okay, we've clearly created a hyperpermeability that's associated with transport and exercise stress. So can we modify that if we change the diet? So something that we were interested in looking at is a fermentation product of Aspergillus oryza, which there's some really nice data in non-equine species that this compound influences hyperpermeability of the gut, primarily through a microbiome-mediated effect. So our question was, if we fed these horses Aspergillus oryza, will that have an impact on leaky gut? So what you're looking here, the panel on the left, this is the same one I showed you before, where we have this increase in iohexol following that transport and exercise stress. And if you look at panel B on the right, these open circles, the top line there, those are the same horses. These are the horses that were exercise stressed after 30 days of receiving this Aspergillus compound. So this is really nice evidence that we can block the effects of exercise and transport stress by altering the diet. So some take-home messages in kind of terms of what we've been talking about the last 40 minutes or so is that the gut isn't just about nutrient absorption. It has a much broader role to play here in terms of the overall health of the animal. So the gut is a critical player in immune response and inflammation. And the microbiome is really orchestrating, I think, a lot of those roles that are outside just nutrient absorption. I think we've also made the point that some of the typical type activities that a horse can go through in a normal horse life, like transport stress followed immediately by exercise, does have a significant impact in terms of developing hyperpermeability in the gut. We can block that hyperpermeability by provision of a prebiotic, and in our case, we looked at Aspergillus. But the broader questions here, I think these data, like much of the studies that we would do, they raise more questions than answers. So some of the questions that come out of this is, well, what is the impact of fitness, right? Our research horses have lived at the research station, many of them for more than 20 years, and they're basically couch potatoes. They don't have a lot of exercise except walking from the water trough to the hay bale and then back to the water trough again. So these are sedentary horses. So would we have had the same effect if these horses were fit? I don't know. So there's I think a lot more questions here than answers, but I think what these data do for me is that they compel us to ask those questions. I think knowing that we can stress horses to the point of experiencing gastrointestinal hyperpermeability just by living a normal horse activity life, it compels us to start asking these questions to see if there are things that we need to do to improve our management to reduce the impact of leaky gut syndrome in horses. So that's all I have for you today, and I thank you so much, and my email address is there. I welcome any questions at any time, and I thank you so much for your attention.